Hi everyone, we are starting Unit 1 and this is going to be focused on post-translational modifications. And so for the devotional for this unit I pulled it from 1 Peter 4:10 through 11. Each of you should use whatever gift you have received to serve others, as faithful stewards of God's grace in its various forms. If anyone speaks, they should do so as one who speaks the very words of God. If anyone serves, they should do so with the strength God provides, so that in all things God may be praised through Jesus Christ. To him be the glory and the power forever and ever. Amen. So God's given us all a lot of different gifts, and we should use those. It's not just the gifts, talents that you've been given, it's the when, where, and how you use them. Those of you that had me for developmental biology know that we have kind of a motto. It's not the genes you have, it's when, where, and how much you use them, essentially. So this course is going to be a little bit different. We're not really focused on gene expression quite as much as we are in developmental biology, but instead we're still focused on the activity of proteins and the activity of cells and how that's regulated. And so we're really going to start that key aspect here in Unit 1. And that really is true for our gifts from God as well. Our gifts from God are beautiful and an amazing thing, but they're just a waste if we don't use them. And when we use them, we need to use them the right way. Just like the proteins we make, the proteins take a lot of energy to make in our cells. And so if they're not used, then that's just a waste. But worse yet, if they're not used correctly, then that can actually cause major issues. So you all have amazing gifts and talents. Some are different in some people than others. But find those, embrace those, thank God for those, and use them. Awesome. All right. So, learning objectives for this unit. After this, I hope you'll be able to understand and describe the various properties of a cell, understand the need for regulating cell activities and the various ways the cells can do that, explain why post-translational modifications, the key aspect of this course really, are important to cell regulation, and then finally understand protein phosphorylation, kinases, phosphatases, and how they relate to protein activation. And a lot of these last couple of points here, we're going to continue on with both synchronous and asynchronous in future videos for Unit 1. All right, so Unit 1 does have a worksheet and crossword. So this is an individual worksheet. Unlike all the other units, the worksheet is actually not due at the beginning of the unit. It's kind of more like our mixed group problems due at the end. But this one is done individually. So this is just there to help you kind of think through a lot of the content and apply it and just make sure you've understood it pretty well. So that worksheet for Unit 1 will be due towards the end of the unit. The due dates are on Canvas. Unit 1 crossword actually is mandatory as well. So there's a lot of terms that we're going to use throughout this course, and I think this is a pretty good review, or way to review a lot of those terms that you should probably know from other courses but may not have thought about in a while. And so make sure you do the crossword for Unit 1. I've given you hints as to what pages to find all those definitions and things like that on the crossword. So it should be a fun, doable exercise that kind of allows us to all be on the same page as we move forward without me just spouting off definitions for you. Reminder again that the other crosswords, basically for the other 5 units, Unit 2 through 6, you have to finish 3 of the 5. So you have 2 units where you don't have to do a crossword, but Unit 1 is not that one of those options. You have to do the crossword for Unit 1. Alright, so obviously here we have a cell, and I just put this up to kind of give you an idea of what we're basically talking about in this course, and the size difference. So prokaryotic cell, you guys all know, has no organelles, no nucleus, hence the term prokaryote. Pro means pre-karyote nucleus. Eukaryote with a nucleus, all the other organelles. We're going to be talking a ton in this class about not only the function of these organelles. In fact, we're not really going to talk about the function directly, but we're going to learn about that in all the other aspects of the course. But we're going to talk about how proteins get to the right places in here, how this cell moves, all those aspects that it's involved. So I want to pause for a second here and just have you go ahead and write down on a sheet of paper or just brainstorm, what are some of the things a cell has to do or be able to do for cellular life? Right? What are some of the critical aspects required for cellular life? See what you come up with. All right, so hopefully you're able to think of some of those important things. The reason I have you do that is really because I want you thinking about this and not just blindly listening to a video, because I think your learning is going to be much better if you're thinking about it and answering questions, and really involved actively in your own learning, similar to what we would be doing in class. And also because really I want to tie together a lot of the aspects that you've learned in previous biology courses to what we're learning about here. So a lot of this is going to be review, but we want to kind of think about all those things so that we can put them into this more detailed context as we go. So what are some of those things that a cell needs to do? Well, it needs to be able to get energy and use energy. And so this isn't what this course is going to be all about, but anabolic reactions are going to build up things, but in order to build up molecules, basically you're going against entropy, it takes energy. And so energy is created when things are broken down. So digestion, cellular respiration, all those things happen. And the sum of that is metabolism. And so together, catabolic reactions basically give us energy in the form of ATP that can be used to do cellular work. And so ATP is important, and that's what we've often heard about, but GTP is just as important. If you think about it, these are the exact same things. This is just the adenosine, and this is guanosine and triphosphate. And so basically they're the same idea. Both can be used as a nucleotide, or both can be used as an energy source when you go from triphosphate to diphosphate. But we're not really going to talk about the anabolic or catabolic reactions. That's more advanced biochem. We are going to talk about the other aspects that these energy molecules are used for, such as cell movement, cell signaling, and regulating all the cellular activities that need to happen. But the cell absolutely needs that energy, and we're going to also talk about how the cell can use the energy efficiently to do these things. Another thing about cells is they're dynamic. So what do I mean by that? Well, that means cells are constantly changing, and they're changing in many different ways. Cells are constantly moving, right? So you can see here that we have a cell that has been grown in culture, something we'll learn about in the lab, and over time that cell is continuing to grow and move. And these are phylopodia. These are kind of long extensions, search parties trying to figure out where that cell should grow. And over here they've created a chemoattractant, so this cell has to respond to that chemoattractant and turn and grow / migrate in that direction. So we'll talk a little bit about cellular movement, how the cytoskeleton regulates that, and so forth. So let's take a look at a couple of these videos of cells moving. There's some pretty interesting aspects along the way. So this is just cells in a dish, and you can see they're constantly moving, but some of this seems like pretty random movements. We actually know now that these cells move much more dramatically in very interesting ways. So let's take a look at another video that really kind of shows this in depth. I love this video because it really shows how cells are actually kind of shapeshifters. So this is a 3D image of a cell using modern techniques that are quite amazing. We've always known that cells move, right? And we'll actually watch this video later on of a neutrophil kind of chasing this bacteria, so it has to be able to know where that bacteria is going in a way. But cells are actually much more complex than that. This is a cell moving through the extracellular matrix. And it's got to move, it's got to get out of the extracellular matrix, and there's constant moving and changing of the membrane and all kinds of things like that. And so we're going to talk about how the cell does that. How does the cytoskeleton allow the cell to move in a very distinct direction? How does the cell know what direction to go? How does it respond to cues from the outside world? How does it kind of have this shape shift, and how does that relate to cellular function in that regard? All right, another thing that's dynamic is movement inside the cell. So we saw the video on Monday where we had our cute little guy that had the 2 feet, was walking and pulling organelles along. There's all kinds of things moving in the cell, and not only are they moving, but they have to get to the right destination and so forth. And then finally, dynamic is chemical reactions. Things are changing all the time. The protein content of the cell, the metabolism, the metabolic changes, and the metabolites that the cell is making and so forth. So we're talking about millions and millions of different reactions happening at all the time. Another basic property of cells is that they have to be able to respond to the environment. right? So how do we regulate these millions of different things going on? How do you regulate the cell movement? Because not all cells should move, right? Not all cells should be undergoing the same chemical reactions. We certainly know that not all cells should express the same proteins. That would mean all cells are exactly the same, right? Differentiation happens because of gene regulation, because which genes are turned on. That's what makes your muscle different from your neuron. But it goes well beyond that. It's the metabolism, it's all the metabolites being made. So how does the cell know what to do? Well, in a multicellular organism especially, you have to respond to the environment. So the cells are cooperating in kind of doing the right thing in a combined organismal manner. But this is true for even simple single-celled organisms. They have to react to the environment, so they're going to where nutrients are, moving away from where toxicity elements are, and so forth. So all cells have to respond to stimuli. This is basically the thrust of Chapter 4, where we're going to talk all about ligands and receptors, signal transduction molecules, so the secondary pathway, and then kind of the responses. But we're going to get into a much kind of heavier detail in that regard. This is how cells respond to stimuli. Cells also have to undergo self-regulation, right? And so in order to make all those other things happen, they have to regulate a lot of different things. But one of those things is cell division and growth. And in fact, in Chapter 6, we'll talk about how cancer happens when this loss of that regulation occurs. But cell division and growth isn't just happening in cancer or not cancer. This is a huge aspect of development and everything else. And for example, or an example of cell regulation, cells grow at different rates throughout life, right? So when are cells growing faster? When you're young or when you're old? Well, hopefully you were growing faster when you were young than you are now that you're an adult. And here's a good example. So my favorite biological example is Ethan. And this is back when he was 4. We took him to Disneyland, and it was on his 4th birthday. He was amazed that everyone there knew his name. He forgot we'd kind of put this button on him, and Mickey and everyone is very well trained to always say, "Happy birthday, Ethan." So he just had a great time. It was awesome. But Ethan, at birth, was 9.5 pounds and 20 inches. Right? So he was actually a pretty big baby, much too big if you ask my wife Beth. But anyway, he was a happy, healthy boy, which is great. By age 4, at this birthday picture here, he was 40 pounds and 42 inches. So essentially what that means is that in 4 years, he had quadrupled his weight and doubled his height, essentially. Right? So if he continued at this growth rate, where he could quadrupled his weight every 4 years, doubled his height every 4 years, by your age, which I'm assuming is somewhere around 20, by 20 years of age, Ethan would be 9,728 pounds and 53 feet 4 inches tall. Right? Obviously, that's not the case because our growth rate slows down. In fact, Ethan is now 15, almost 16, and if his growth wasn't already slowing, he'd be more than 2,000 pounds and 25 feet tall. So cells are very much regulated along the way. And obviously, Ethan is a lot bigger than he was at 4, but he's not 2,000 pounds nor 25 feet tall. Cells also need to regulate metabolism. They need to regulate how much energy they're making and how much energy they're using. Sometimes a lot of energy is needed and other times less, and it really depends on the environment and so forth. They need to regulate their activity so they're doing the right thing for the organism at the right time. And then also, cells have to undergo differentiation. This is a big part of being multicellular, is because our cells end up with different functions, specialized functions that work well together as a whole. Neurons do their thing, red blood cells do their thing, muscle cells do their thing, leukocytes do their thing, and so forth. All right? This is talked about a lot in developmental biology, how this happens, but just kind of understand that this is really, really important to cellular function and ultimately is critical to the multicellular organism being alive. All right, so what I want you to do is pause again and kind of on that brainstorming paper, think about some of the ways that a cell can regulate its behavior activities. So I've just talked a lot about how a cell is dynamic and a cell is constantly changing and how those changes are very important that it's able to do that. And in fact, cells have to react very quickly and change their behavior, their movement, whatever, very quickly. But it's also extremely important that those are regulated. So what are some of the ways that a cell's activities are regulated? All right, so hopefully you were able to get some ideas down. At this point, we've really talked about the first two objectives already, describing the various properties of a cell, understanding why regulating cell activities is really important to maintain and regulate those dynamics and so forth. So now we're going to kind of move a little bit forward and talk a little bit about how that is regulated. Post-translational modifications are going to be a part of that. All right, so let's think about this for a second. How can a cell regulate its activity? Well, there's various levels. Ultimately, and proteins aren't the be-all end-all. There are a lot of RNA enzymes, there's a lot of siRNA, there's a lot of things that regulate a cell, but ultimately, oftentimes, to a great extent at least, we're talking about regulating the activity of a protein, or the protein, the combined set of proteins in the cell. So the first level of regulation that you've learned a lot about already is transcriptional regulation. Right? So what genes are being turned on? Which proteins are being made? So at the gene level, we're regulating a cell's activity by regulating what proteins are being made by what genes are being turned on. And we talk a lot about this in depth in developmental biology, and in fact this is the key aspect of how a cell differentiates. This regulation is extremely, extremely important, right? But it's not the only regulation. So there's other levels of regulation that we're going to talk about. So once you have made the mRNA, once the gene is transcribed, you have splicing. And if you have differential splicing, you can take the exact same gene and make two or many sometimes different versions of that protein by whether or not certain exons are included or spliced out and so forth. So that is a key level of regulation, and there's a lot about that that we still don't completely understand, but we know it is highly regulated which versions of the protein are being made. And in some cases, two splice variants of the exact same gene create two proteins that have the exact opposite effect. Right? For an example, BCL2 we'll learn about, and this is a protein involved in apoptosis. One splice variant causes apoptosis, and one splice variant prevents apoptosis. And you can maybe start to imagine a little bit why, because the function of this guy here is going to be very similar to this, except it's missing this activity. So if this activity might cause apoptosis, this activity might actually block it because it can compete. If these two parts here are what's binding to the receptor or binding to some other factors that are allowing it to do its job, this can compete with it but doesn't have the actual activity. But anyway, that's kind of a long story for another day. Differential splicing is a really important aspect of regulating what a cell is doing. You can also regulate mRNA stability. Obviously, the more stable an mRNA is, the longer there is for translation, and therefore you make more or less protein from the mRNA based on its stability. And this can also lead to translational regulation. So even if the mRNA has the exact same stability, you can regulate the ribosome and how much is being translated. So these two things, 3 and 4, are really kind of giving a bit of regulation on how much of that protein is being made. And then finally, and really important for this course, is post-translational regulation. So even once the protein's there, you can actually turn that protein on or off, essentially, through post-translational regulation. We're going to learn all about phosphorylation, how the proteins fold differently based on its post-translational regulation, and different quaternary interactions. So some proteins are only active if they're bound to a cofactor or another protein, things like that. So this is post-translational regulation. The protein is already made in the cell, but you can turn it on and off, essentially. Right? And so these are going to be a big part of our signaling kinases and things like that we're about to talk about. All right, so how long do you think this whole process takes? Let's go back to kind of this part, the transcriptional regulation, right? So in transcriptional regulation, we have our DNA. It gets transcribed to mRNA, and then the mRNA gets made into protein. How long do you think this takes to start, to basically make one protein? All right. If you said that it takes about a minute, you're right. In fact, sometimes it can be a lot faster than that. And this is actually quite amazing when you think about it. All right? Think about this for a second. The process of finding the right gene, so the RNA polymerase finding the gene start site, becoming activated, so gene regulation is included in this. and then unzipping the DNA temporarily, RNA polymerase moving along. In most genes at this point, remember, you have huge introns. The introns sometimes are 10 times long or 100 times longer than the actual coding part itself, even more. So the gene itself is usually tens of thousands of nucleotides long. So the RNA polymerase has to transcribe tens of thousands of nucleotides. Then you get the RNA modifications, right? The 5' and guanosine flipped cap, the poly A tail, the splicing. And then not only that, but once you have the spliced one, you're going to the ribosome and the tRNA is coming in. You're linking the amino acids together. So the fact that you can make a protein from start to finish in less than a minute is absolutely amazing, right? This is way faster and more complex than any factory we have here on Earth. And the cell is doing this with thousands of different proteins at a time. Right? So I'm not at all trying to take away from how amazing this is, and I hope you really appreciate how amazing that is. But that 60 seconds is an eternity for a cell. If a cell basically had to rely on regulating itself by making a protein when it needed it, and then degrading it when it didn't need it, and then making it again when it needs it, and degrading it when it doesn't need it. So in other words, if a cell did not have a way to turn a protein on and off without making and degrading it, the cell's ability to react to the environment would basically take an eternity in a cellular life, right? So we need a different way of regulating those immediate dynamic changes that a cell has to make. Again, this is not saying that gene regulation is not important. It's absolutely critical, but gene regulation kind of does more of the differentiation aspect and kind of the longer term differences. The short term changes that a cell reacts to have to be reliant on something that's much faster. In addition, not only do we need something that's faster, but if you think about it, it's not possible to make proteins based on the cellular environment and then degrade them. Because if we think about a way a signaling cascade has to happen, the ligand binds to the receptor. That receptor is not inside the nucleus. So we have this signaling cascade of proteins. And then that causes gene expression. Well, if this ligand bound to the receptor, if these guys are always on, you're always getting gene expression changes. But if the only way to turn them off is not have them there, then you cannot create this signal and turn on a new gene because your signaling cascade molecules aren't there. This is like a bucket brigade, the bucket brigade that used to put out fires. Right? So you would have the first person kind of grabbing the bucket, then you would pass it to the next one and pass it to the next one and pass it to the next one, right? And then finally it gets to the end and you dump that out. If any of these molecules are not there, if any of the people in that bucket brigade are not grabbing that bucket, you can't get to the end. And so if the gene expression changes were dependent on proteins already there, but those proteins could not be turned off, this wouldn't work. I know this is a little bit hard to explain, but pause and think about it for a second. We have to, here, let me try this one this way. We have to have these proteins already in the cell for this to be possible, but they can't be on at all the time. So in other words, these proteins have to be made always and available to transduce this signal, but they have to normally be off, and this has to be able to turn them on. And so we need a way to regulate these things that goes beyond gene expression transcriptional regulation. And so with that, we get down to these levels here, and for the most part, especially with the example I just gave, where the protein has to be ready to do that signaling translation, that signaling cascade, we need a way to turn it on and off. And that is our post-translational regulation. So modifying the protein that's already made to turn it on and off. That is going to be rapid, it's fast, because you don't have to make and degrade the whole protein, and it's needed because there's many times we need the protein there ready to be turned on, but we need it to not be on by default. We need it to be off by default and then turned on. Right? So the example that I give for this is a light switch. Right? This is my analogy. So let's think about this for a second. I don't want to have to rewire this whole room in order to turn on this light. And once the light's on, I don't want to have to cut all the wires to turn it back off, and then rewire the room to have a light, and then cut all the wires to turn it back off. That would be what's happening to a cell if, in order to have a function, it had to make the proteins to have them on, and then degrade them to have them off, and then make them again to have them on, degrade them to have it off. That would take a really long time just to turn the light switch on and off. But if I walk over here and I just flip the light switch, I can turn the light on and off with that switch because everything is still there ready to go, but I modify, in this case, the electrical current, but I turn it on and off. That's what our post-translational modifications are doing. Alright, I know this has already been a pretty long video. I'm going to stop it here. Again, you can kind of come back to this anytime, but we're going to stop here, and then the next video is going to really focus on these post-translational regulations. So that way you don't have to scan through the whole video to get to this second part if you want to come back and review later.